The kidney is comprised of three primary sections: the renal cortex, the outer shell, the renal medulla, the cone-shaped tissues, and the renal pelvis, which is the cavity connected to the ureter to facilitate the transportation of excess waste. Within both the renal cortex and the renal medulla, nephrons exist within them as seen through this diagram. These perform various functions including the production of hormones, osmoregulation, which is a regulation of water and salt concentration, which both contribute to blood and volume pressure, the regulation of acid-base balance through the transference of hydrogen ions and bicarbonate. However, their primary function is to excrete and filter the metabolic wastes of urea, which is a nitrogenous waste, ammonium, creatinine, uric acid, and many others. Firstly, the nephrons filter blood through the glomerulus as seen here in the orange diagram. Firstly, the nephrons filter blood through the glomerulus as seen in this orange diagram, which receives unfiltered blood from the afferent arteriole. The glomerular intakes the blood and conducts the process of pressure filtration as the hydrostatic pressure of blood, which is the pressure of blood in a confined space, results in the forced filtration of water and other small molecules into the Bowman's capsule as seen here, otherwise known as the glomerulus capsule, and then into the tubules of the nephron. The filtered molecules are known as glomerular filtrate. The unfiltered larger molecules then exit through the efferent arteriole, which connects to and supplies the network of peritubular capillaries, which are not visible in this diagram. These peritubular capillaries travel along the nephrons to allow tubular reabsorption of sodium chloride, water, glucose, and amino acids to occur, whilst the further allowing the tubular excretion and secretion of potassium ions, hydrogen ions, and many more. The peritubular capillaries then connect to the renal vein, which recycles the nutrients back into the body. However, for pressure filtration to successfully occur in the glomerulus, it must achieve a certain degree of hydrostatic pressure of approximately 60 mm of Hg or mercury. Once the filtrate consisting of water, NaCl, amino acids, glucose ions, and other molecules, it is then transported to the proximal convoluted tubule, as seen here in the green. This is primarily used to perform tubular reabsorption to the peritubular capillaries. Within this process, multiple molecules are reabsorbed passively, as seen through these purple dots, and actively with energy input. The majority of glucose and amino acids are reabsorbed into the blood via this process at the PCT. In addition to glucose and amino acids, ions are also reabsorbed at the proximal convoluted tubule, with approximately 60% of sodium chloride being reabsorbed here actively, with some additional excess chloride also being reabsorbed. The reabsorption of ions leads to increased osmolality, which are solute particles within 1 kilogram of a solution. Subsequently, as solute osmolality increases, there must also be an increase in osmolarity, as water follows the concentration gradient of solutes. Subsequently, the increase of osmolality gradient formed by the active and mainly the active transport of ions, which are unfortunately not visible on this diagram, creates a osmotic gradient within the PCT and the peritubular capillaries, which allows the immediate reabsorption of water at the peritubular capillaries and the eventual reabsorption of water at the loop of Henle through the processes of osmosis. The majority of water is also reabsorbed within the PCT due to the abundance of aquaporins located in this area. This allows for the passive transport to occur in this location, in which approximately 67% of water is reabsorbed. In addition to this, the majority of bicarbonate, which is HCO3, is also reabsorbed at the PCT, which plays an important role in the balancing of blood pH levels. The proximal convoluted tubule also contributes to tubular secretion of acidic waste, as certain products, including penicillin, ammonia, creatinine, and histamine, are all secreted into both renal tubules for eventual excretion. The loop of Henle. After the filtrate has undergone both active and passive transport via reabsorption, it is then transported into this loop as seen here, which is known as the loop of Henle. The loop of Henle exists in the medulla of the kidney, defining the boundary between the renal cortex up here and the outer medulla down here. The loop of Henle consists of two primary parts, the descending loop in green and the ascending loop in purple. Within the ascending loop of Henle, it is permeable to ions but not water. The remaining sodium chloride, which has not been reabsorbed within the PCT, is transported once again to the peritubular capillaries here. Within the lower part of the ascending loop, NaCl is reabsorbed via passive diffusion, whilst in the higher regions of the ascending loop, NaCl is reabsorbed via active transport. This is due to the evident thickening of the ascending limb, as you can see here. Therefore, as diffusion is a much faster process due to not requiring an energy input, the majority of NaCl is reabsorbed in the lower areas of the loop of Henle, as can be seen in the diagram. The unequal distribution and reabsorption of NaCl between the lower and higher parts of the ascending loop results in a great concentration osmotic gradient, as there are greater concentrations of NaCl further down the medulla and further down the ascending loop of Henle. Unlike the ascending loop of Henle, the descending loop is permeable to water and non-permeable to ions. Within the descending loop of Henle, water is reabsorbed for the second time after the PCT. As the descending loop is semi-permeable to water due to the presence of aquaporins. Subsequently, this allows for the passive transport of water to be passed through the descending loop of Henle to the peritubular capillaries. Furthermore, due to the existing osmolarity and concentration gradient of the medulla, and the concentration differential of NaCl within the ascending loop, the majority of water is reabsorbed in the lower locations of the loop of Henle due to the concept of osmosis, which states that molecules tend to pass through a semi-permeable membrane to higher concentrated solutions, which in this case would evidently be the lower parts of the loop of Henle, or the lower parts of the descending loop of Henle. Furthermore, the concept of the countercurrent mechanics of the descending loop of Henle further contribute to this concentration gradient, as once H2O is reabsorbed from the descending loop of Henle into the peritubular capillaries, the differential between the osmotic gradient slowly begins to increase in the gap, and subsequently, due to osmosis wanting water to go to areas of greater concentration, this will continue to occur as the gap begins to widen, which creates a cycle otherwise known as the countercurrent mechanism. For the distal convoluted tubule, which is seen in this purple area, the distal convoluted tubule is primarily responsible for tubular excretion and secretion. Within this process, the positive potassium ions, acidic hydrogen ions, and urea are passed from the peritubular capillaries back into the distal convoluted tubules for eventual excretion. In addition to this, tubular reabsorption also occurs as the remaining sodium chloride and bicarbonate are then reabsorbed. However, these amounts are very minuscule. Subsequently, as DCT is primarily used for the tubular excretion of positive potassium ions and hydrogen ions and urea, it generally plays a large role in the balance of pH levels of blood. The high concentration of mitochondria within the DCT further enables the active transportation of certain ions, which aids the entire process of tubular secretion. Finally, for hormones, there are three main types. There's aldosterone, ADH, and ANH. Firstly, aldosterone. For aldosterone, the juxtaglomerular apparatus, which is located nearby the glomerulus, acts as a quality control mechanism to maintain levels of blood pressure via sodium reabsorption, which can be seen here, and also in the PCT. When a person's blood pressure is too low, known as hypotension, the juxtaglomerular apparatus is triggered to release renin, an enzyme which converts the hormone angiotensin released by the liver into angiotensin 1, which is then converted into angiotensin 2. Once angiotensin 2 is produced, it then interacts and stimulates the adrenal cortex, which are located above both kidneys, to secrete the hormone aldosterone. Aldosterone then interacts with the receptors of the renal cells located in both the DCT and the PC2 to activate the production of ion channels within them, and also the aquaporins, which allow greater passage and reabsorption of NaCl and water. Conversely, another sodium regulating hormone is atrionatriuretic hormones, which otherwise known as ANH, which is hormone secreted by the atria, which is located in the heart. It is secreted due to the stretching of the atrium as a result of increased blood volume, otherwise known as hypertension. The ANH then inhibits renin secretion and therefore aldosterone to limit the activation of these ion channels located here, and therefore inhibiting the reabsorption of sodium and water via osmosis to limit the blood volume. When excessive hypertension occurs and urine needs to be hypertonic in comparison to the blood flow, ADH is known as antidiuretic hormone, and it is released from the posterior pituitary gland. In the presence of ADH, the collecting duct over here becomes more permeable to water, allowing the urine to become hypertonic, whereas in the absence of ADH, the collecting duct here becomes less permeable to water and remains hypotonic. Subsequently, the primary function of antidiuretic hormones are to regulate the concentration of urine and therefore the blood pressure and volume. Thank you.